So this slide is going to show you the basic framework for data generation and risk assessment under REACH. And of course, you have two major parties. Other people are involved, but these are the two major parties: the registrants on the one hand side, and the authorities, such as the BFR, other authorities in Germany, and of course the European Chemicals Agency in Helsinki. So what do registrants do if they work according to their obligations? They are obliged, according to the tonnage marketed, in which their substances are marketed, they are obliged to provide standard information, which on the toxicology and is mostly still in vivo testing. There are some in vitro standard information already defined. They are going to provide this data, and the data are then collected in the registration database. As of October now, this is from the ECHA website. We have received over 90,000 registrations on over 20,000 substances and from over 14,000 companies. So talking about big data, here it is. You see that. Now, I talked about the standard information, but one essential paradigm of REACH is also that animal testing should only be used as the last resort. If you have another way of dealing with the requirement, please do it without animal testing. And that process, there are some other ways to do it, but the main process to use in silico methods for that purpose is called adaptation or walving, meaning adapting the standard information requirement to something different. So they can all also go this way, use whatever, non-guideline tests, weight of evidence, QSARs, read across. And again, the data also go to the registration database. They are also obliged to do what's called chemical safety assessment, which includes hazard characterization, exposure assessment, risk assessment, quantitative risk characterization in the form of health-based guidance values. They are called DNLs, derived known effect levels, or DNLs, under REACH. And as a result, you get some, for instance, the DNLs, some risk assessment figures that go here. And you get also, you might get a classification for hazardous properties that goes into the classification and labeling inventory, another database, in which currently data on about 145,000 substances is collected. So at this point, actually, our presentation could stop because registrants should do their job. They should judge the hazard of the compound or the risks, take measures, and that's it. Only it doesn't work, of course. So you need the authorities to evaluate, I'm sorry, to evaluate whether the registrants did their job properly and provided all the data that are needed. So we check this, and if we find there's something missing, a data gap, we issue an information request to the registrants. Sometimes they identify it themselves. And so for the higher tier tests involving vertical testing, they need approval by the authorities before they go ahead. So the testing proposal. But the authorities, of course, concentrate also on high-risk substances, or try to do that. It's difficult because of lack of data often. And we implement screening procedures, and we're going to talk a little about that afterwards. And as a result of that screening, we can, for instance, find out that more evaluation is needed in terms of data gaps. Or we do a chemical safety assessment ourselves, which may then result in classification and labeling, or in further regulations. There are some processes under REACH, such as authorization or restriction, the identification of so-called substances of very high concern, or, of course, you can take the information you gathered here and do some regulation in other frameworks that are there for chemicals, for special, I don't know, cosmetics, toys, and what it is. So about screening, a few slides. We heard yesterday already, for instance, the US colleagues have a concept involving also high throughput screening and NAM methods. Also in the free literature, you get papers nowadays. So in silico methods are used for screening. And I'm going to show you a little, very hot off the press. This is from a presentation that was held last week at the ECHA screening workshop. And the authors were so kind to allow me to use their slides, but they really told me to tell you this is work in progress. This is not the final result, but otherwise, I wonder if I've got the clearance here. So what they essentially do is they take process data to classify registered substances into 4 into one of 4 bins. You probably cannot read it from behind. So it's just a basic prioritization step. Substances with low priority, substances with a high priority for risk management, where we already know there's a risk. And also substances with a high priority for data generation, where data gaps have been found. And there's this large area where they are uncertain where to put this compound. And so this is step 1. And in the next step, then they try to estimate the likelihood of exposure of certain groups and also the likelihood of hazard for those compounds. And in the next step, then they make another 3 bins, which means these must be verified, low priority, or you get a pre-check verification, which ECHA does, or this is further processed by the member states in a detailed manual verification of the priority setting. So that's the basic scheme. And for step 3, or to go from step 2 to step 3, no, I'm sorry, this is actually wrong. This is step 2, of course. I go back. Step 2, to calculate the hazard likelihood and the exposure likelihood, they use certain mechanisms, or they're in the process of finding out which mechanisms work best. For certain substances with well-known chemistry, they can assign it based on that chemistry. They may use grouping. You know, if you have a substance in one of the priority groups, and now from the uncertain pool, you find substances that's similar, you make a grouping, and then you conclude that this substance also might have a high priority. But the most interesting part from the in silico perspective is mechanism C. They use screening algorithms, and they use all kinds of algorithms, exposure indicators, but also hazard scenarios. I can give you the details here, but these are really QSAR based on some regulatory lists, very complex grouping and screening algorithms. It's very interesting what they do. In the end, they might even have to prioritize within those bins. So that's how screening is done, partly big data, partly manual and foot. So next area is information generation. You know, any type, whether you provide standard information or you use adaptation, there are some fundamental requirements. And these are, yesterday we heard mentioning the scientific method. These are essential features from the scientific method, basically. So the relevance of the information needs to be checked. You know, some information may only be relevant for certain contexts, certain goals, certain endpoints, and you need to check on them. The reliability needs to be assessed, traceability, reproducibility, repeatability. I think you said replicability, I don't know what the exact difference is. And also the adequacy for the regulatory endpoint must be established. And the particular point about the standard information is that for all higher tier endpoints, this information needs to allow for classification and labeling and or risk assessment, and that's quite a hurdle for some of the methods in the QSAR field, which only can do part of this. And they should cover, if you think of a repeat dose toxicity study, which has all kinds of toxicities, liver, kidney, whatever, any model should cover the complete set of key parameters or any combination of models to be able to replace that standard information method. And of course, documentation is absolutely key. And you should use it to be transparent. It should be adequate and reliable too. You should use standardized templates for communication. It also can structure your own evaluation workflow. It's a checklist of things that you should consider, and also it allows us regulators to easily access the data because we know where to look for certain information. And last but not least, it assures that this information can go into databases and can be further mined. Yeah, maybe this is the most important slide. I don't know, less scientific, but most important. It's a bit about the mindset of regulatory toxicologists and authorities. Now, same as you, we all come from diverse backgrounds. In my group, we have people, we have a veterinary pathologist, we have biologists, biochemists, chemists, I think food chemists also. And they all have their special expertise on some aspect. But of course, they have, or should have at least, a good general overview of the regulatory toxicology field. They know about all the endpoints, but they are not an expert in every endpoint. You know, we don't have an expert who's specialist in endocrine disruption, liver toxicity, and PBPK modeling. At least we don't have an expert like this. But these are different people. You know, so to guarantee that we are working on the highest scientific level, that we consider all the up-to-date science, we need mechanisms for the experts to convey their wisdom to us, you know. And this is done, I'll show you in a minute how it's done. And then you shouldn't forget, authorities are not composed of regulatory scientists alone. You have risk managers, legal staff, you have people that are not trained in science even. And you need to sort of find a way to clearly communicate what you're doing. Also, so you need a clear assessment, standardized assessment framework for communication with all the stakeholders, also NGOs, the public, whatever. You need a consistent evaluation practice. You don't want that your one regulator, based on the same data, concludes the substance is, let's say, carcinogenic, and the other one concludes based on the same data it's not carcinogenic. Yes, you know, industry has a right for equal treatment under the law. Also, we need to act proportionately in terms of the burden we place on industry. We cannot tell them to do all the costly tests if there is no visible outcome for them. So that leads me to the statement that authorities, I mean, we are scientists, we're always interested in new science, but from a viewpoint of an authority, we only need new science if it somehow improves our risk management. You know, if the new science is much more expensive, but it doesn't contribute anything considerably new to the risk management in the end, why should we pay money for it? And we are bound to efficiency because we are spending a lot of taxpayers' money. You know? So the process, and we are also mentioned, I think, or someone did, it's really a long process if you have a new method to achieve the regulatory use of it. You start off, you have those new methods, then you gain first practical experience, you need to publish that, you probably will publish it, and then at some stage you think it's ripe enough to to undergo validation for a certain regulatory use. If that happens, if that's successful, you might end up in the guideline, as Andrea said, and there will be guidance documents explaining how in detail this method should be applied, what are the pitfalls, and so on. Also, within silico methods, you probably need some tools and templates that are, again, also validated for the purpose. And if you have all that, then you are ready for regulatory use. And this may take many years. So let's talk about how QSARs are used. QSARs, by QSARs I mean that's the standalone use of a QSAR profiler, not QSARs in combination. I will talk about that later. So one of the key requirements for QSARs under REACH is that the model must be, whatever that is, scientifically valid. We did that already, I think, 14 years ago. The OECD principles for the validation of QSARs were published, and you know them probably, or some of you will. The most important things are a defined endpoint. If you don't really define precisely enough what you're predicting, for instance, if you say, I'm predicting reproductive toxicity, nobody in regulatory field will really understand what you're talking about. Is it about malformations? Is it about a developmental toxicity study? Is it about fertility? Whatever. So you need to be precise. You should have an unambiguous algorithm, which means if person A uses this algorithm, he will obtain exactly the same result as person B. And also, it needs to be understood by the regulator. So, for instance, artificial neural networks may come across as sort of a black box, and that's difficult for us. You need to define the domain of applicability. Also, if you then have a prediction by a model, you need to show that your substance must fall in the applicability domain. Of course, you need performance indicators, appropriate measures of goodness of fit, robustness, and predictivity. And also, you need a mechanistic interpretation if possible. That doesn't necessarily mean you need a full-blown AOP, but you need some kind of hypothesis why your model is sort of, or the descriptors that you use to build the model are sort of relevant for the task. And as I said before, the results need to be adequate for the purpose of classification and labeling and or risk assessment. Now, if you take read across as a technique, that's this is the framework and the REACH. For read across, you need to demonstrate that some substance properties, which are physical, chemical, toxicological, and ecotoxicological properties, that they are likely to be similar or follow a regular pattern as a result of structural similarity. And that can be based on a functional group or common functional groups. Or maybe you have common precursors, or one substance degrades into the other, and so on. But structural similarity is key, and also a constant pattern in the if you have a group of substances in category, constant pattern in the changing of the potency is needed. Of course, the adequacy reliability criteria apply too. But from that, what you see is the vision that some of us have, or some people have expressed, that we will transgress to a more biological read across, or even a biological read across alone without structure similarity. So you have maybe some common AOP, you have all the test, two chemicals act in the same way on your cell culture, whatever, by the same mechanism, but they are structurally diverse. That is currently not possible in REACH. I mean, REACH can be changed if science changes, but this is hard-coded here. You need structural similarity. So for a long time, people did how they chose to do it. But in 2015, ECHA and with some member states developed this read across framework. You might have heard of it. It's essentially a checklist for ECHA assessors, not to forget any critical points, but it has turned in sort of a framework for how you should do read across and a checklist on what you should consider. This is just an example. There are different scenarios. This is an example for a category. So one so-called assessment elements, substance characterization. We struggle a lot with substance ID questions under REACH, just to show that the substance really you talk about is really the one that you claim to talk about. In a category, you need to analyze, of course, the structural similarity, but also the differences within the category, which leads then to you analyzing the link of the structural similarities, but also the structural differences with the proposed regular pattern. You need to see a consistency of effects in the data matrix, or the reliability and adequacy of the source studies. And this is an important point, the bias that influences or might influence your prediction. And with read across, this is particularly relevant. You have potentially you are prone to selection bias, meaning from all of these chemical universe, you now picked 4 or 5 chemicals for read across, but there might be 5 other chemicals which somebody could say they are also similar. Why did you not pick those and only the other 5? So many questions. It's really tricky business, and the assessors at ECHA, if they get a read across classification, they go through the elements and they assign scores from 5, like very good, down to 3, I think still acceptable. I forgot whether it's 2 or 1, which you don't. I think 2 is not acceptable at the moment, but it could be improved, and 1 is really not acceptable also. And the lowest score wins in this case. If one of the elements is not acceptable, the whole justification is not acceptable. But it is, as I said, not only an assessor checklist, but also for people doing read across. It's very interesting. If you're planning to develop read across tools, I can only recommend that you go through this document. So how does it work in practice? What kinds of information do we get? This is based on almost 7,000 registration dossiers from the higher tonnage levels. This is from 2017. ECHA's report for 2018 is not yet there. The blue is essentially testing, meaning the light blue is studies that have been there already before REACH. The blue are new studies, or assumed to be new. And for our interest, it's the orange and the red. So red is QSAR, and you see it's only marginally used. You heard it's different. Criteria are strict, marginally used on its own. And then you have read across, which might involve some QSAR aspects too, but in an integrated approach. And that's for more use. And here you have the most expensive, most enable consuming studies. Developmental talks repeat those, toxicity reproduction. I should say carcinogenicity and toxicokinetics are not strict requirements. They are only toxicokinetics. You are not required to submit any information that you don't have. And carcinogenicity you only are to generate if you have certain indicators, mutagenicity, certain other indicators. So That is the picture. And you see that read across is used far more often than users. And you also see, you don't see that, no, you don't see it here, but we see it in practice, that insufficiently justified read across is really one of the main causes that your adaptation fails. You see the decisions, ECHA, we see the decisions where they think it failed adaptation. And there are other reasons, but insufficiently justified read across is really a big problem in that regard. And you should understand the processes are they have tight timelines. So the registrants ECHA checks, and then they get a small chance, maybe 4 weeks, to improve their read across justification, but they have not more time. Otherwise, they need new testing. So there is some activity going on to improve read across on the industry side, to provide more guidance how to do it. There was a workshop in 2016. It's an interesting document. They analyzed certain decisions by ECHA and found main reasons why a read across was turned down. One reason was, you can probably not read it there at the back, unclear substance identity. It was not possible to ascertain the structural similarity. A lack of sufficient evidence to substantiate the assumptions made within the read across justifications, for instance, mechanistic, or often it's toxicokinetics, read across to inappropriate data. For instance, they read across a study that was not enough for the endpoint where they should have submitted a study, and lack of scientific plausibility. And these people identified some areas for more work. It should be, they said, well, the RAAF, the risk assessment framework is, the read across assessment framework is something for the authorities, and we need to transform that into better guidance for registrants. They also recommended a continuous process of case studies to better elucidate how you do it, and to establish a best practice also for integrating biological profiling and bioinformatics tools. And there is this year, I was there was a follow-up workshop, and you can expect in 2019 a new report where we talk about good read across practice, hopefully. Let's see what comes out of it. The next step now is the integration of in silico methods in larger frameworks of assessment. And I don't know if everybody of you has heard of what an IATA is. An IATA is an integrated approach to testing and assessment, something like this. This is for skin irritation only. You don't have to read it, just to show you there are some steps and it might be complex. So it's an approach for hazard identification, characterization, safety of chemicals. It integrates and weights all relevant existing evidence, including in silico models, grouping, read across approaches. And it guides also the targeted generation of new data where that should be required. So you identify data gaps, and some of the pipelines here lead to new testing as the last resort, as I said. And this should, wherever possible, be mechanistically informed. And we've heard about that. Mechanistically informed essentially means nowadays it would be good to have an AOP to this. And how is an IATA based on an AOP? This is an AOP, molecular initiating event, key events, growing level of biological organization from molecular to whole organism or population level. You see it. And here, this is, I don't know, from 2016 maybe. This tells you what OECD at that time thought where certain types of information could kick in. I'm a little bit astonished. I only noticed it today that in silico, in their view, would only be restricted to the molecular level, whereas I would say you could almost go all the way for some kind of in silico methods. But in silico, standalone profiling methods are probably best placed there. Out of the IATAS, so I should have said the IATAS we saw, they are a framework. You can handle them in a different way, also depending on which information you actually have or prefer to use. It's only a scheme. If you have that information, where does it belong in the scheme, and what do you do with it? Now they invented something that's called a DA, a defined approach for testing and assessment. And that's one concrete way of living in IATA, or a part of an IATA. So in a defined approach, you have a fixed data interpretation procedure, which is called DIP or DIP. And that's, of course, a good thing to automate or to integrate into IT solutions. And of course, you have a defined set of information sources. So it's one particular way of solving an IATA. And I give you an example of a popular one. we have, I should say some more, we have an AOP for quite some time now for skin sensitization, you know that probably. And there's also there has been work on how to lift this AOP and transform it into an IATA. And now there was an OECD project where certain contributors could enter their way of doing it, their defined approaches. And currently, the OECD is in the process of trying to establish a so-called performance-based test guideline for defined approaches for skin sensitization, which is a totally new thing. They found out at some stage they couldn't validate individual methods any longer with this complex and time-consuming schema I told you about.